So you guys are ready for a bit more, or you're tired?
Yes, sir, we are ready.
Yes,
Oh,
sir, we are ready.
very good. That is very encouraging, right? So it is nice that you're ready. So we'll talk again a bit more about the sequencing and how today the sequencing technologies have changed and how we are now into a world of sequencing where everything begins with the start of sequencing. So here we are. Let's listen in a little more. So we are talking of 2001. This is the sequencing of the human genome. So what is a genome? Genome is one full complement of genetic material in an organism or cell. And sequencing genomes is challenging. Why is it challenging? Because there is still no technology that can directly read a full chromosome from one end to the other end, right? Let alone the genome. One single full chromosome also cannot be read from one end to the other end. So what effectively happens is you have to break the DNA into smaller pieces, read individual fragments, and then put these fragments together in the order in which they would appear in the normal single united DNA, right? So that is where the challenges come. We'll talk more about it and show you a diagram. So genome is one full complement of genetic material in an organism or cell, and sequencing genomes is challenging. What is sequencing? Sequencing essentially means to determine the order of nucleotides, ATGC, in a given DNA molecule. And the genome sequencing projects began around 90s. The basic idea of sequencing the human genome arose around 1988. In 1990, it was formally launched as the Human Genome Project, and it took around 11 years to sequence the first genome. So you can understand the complexity and the challenge involved at that point of time. The only sequencing technology available was Sanger sequencing at that point of time again, right? So, and the question then is, why are we sequencing the genomes? Well, we are sequencing the genomes because we want to understand the basic composition of, let's say, a human genome, a chimpanzee genome, a rat genome, a mouse genome, a cow genome, an Arabidopsis genome, let's say a Neanderthal genome for that matter, a mammoth genome, and so on and so forth. So we are doing sequencing because we understand the basic composition. DNA is the blueprint, and blueprint has been very strongly conserved. So we want to see what is there in the blueprint. Then, of course, we also want to understand the basic composition. We want to do what is known as comparative genomics. Comparative genomics means you're comparing genome sequences, and you could do this comparison intraspecific within the species. You could do it interspecific between species, right? So when you do between humans, you're doing intraspecific genomic comparisons for one to identify variations in the DNA that are responsible for disease phenotype or that can push you into a disease. They are likely to push you to a disease, right? You could also do this kind of comparative genomics to identify the origin and descent of humans, the direction of movement of human population, human civilization, how various areas of the earth were occupied, where the original, how the invaders came, and how the original people were then pushed out, and so on and so forth. But more importantly, we are doing these comparisons for biomedical reasons to identify regions in the genome and the variations in the genome that result in diseased phenotypes. So that is basically the most important and biomedical application of human genome. I'm showing you two people here. This is Francis Collins, the director of Human Genome Project, the government-funded 10-year-long project led by the United States of America, NIH, and basically, almost 50 other countries and some of the best brains of the world involved. The other person here, which is a parallel historian in the Human Genome Project, is what is known as this person is called Craig. Was initially part of the Human Genome Project, but then he broke away from the Human Genome Project on certain key issues of how to approach these solving the problem. And that is when he formed his own company called Celera Genomics. He was a believer that we had enough trained computational scientists and enough trained computers to be able to sequence the human genome with what is known as the whole genome shotgun sequencing strategy. Now, we'll not go into the strategy for now, but you should know that there are two strategies that can be employed for sequencing the genomes. One is what is known as whole genome shotgun, and I'll show you an idea of how that is done. And then the other is what is known as the hierarchical shotgun. And we'll talk of that in a moment now, or maybe tomorrow a bit more, right? Because this is something that you need to know. But anyways, if you look at how to sequence a genome, the steps are still the same, whether you talk of 40 years ago or even today. When you're sequencing a genome, the first thing is to break the genome into smaller fragments, right? Now, these fragment sizes can be different depending on which sequencing technique you're using, but the idea is still the same. You are basically taking a DNA, you're breaking into smaller pieces. Each of this piece is sequenced, and once it is sequenced, these pieces are then assembled back so as to be sure that they are in the same order as found in the original genome. So that is the challenge that is there. The technologies that are available for sequencing the genomes now today, uh, beginning of 1977 until 2001, Sanger sequencing was the only one that was automatable and was used. Sanger can give you more than from a single sequencing primer up to 900 bases of sequence, right? So the fragment cannot be more than 1,800. 1,800 meaning you could sequence half part from here and half part from the other end. So therefore, these fragments could be uh, roughly around 1.8 kb, 1.9 kb in terms of size. Then you have now what is known as the after 2001, this Human Genome Project took around 10 years and $3.2 billion to sequence the first human genome. And we realized that this is not the technology that can take us to precision medicine, because if it is going to take $3.2 billion to sequence and 10 years to sequence, then of course it is one, it is not affordable. Second, the patient may not have that much time to wait until 10 years for a remedy, right? Even if, any disease, 10 years is a very long period to expect to, gain first the only the information, and then the second part is understanding. So therefore, it was realized that Sanger sequencing, while its crowning glory is the sequencing of the human genome, it cannot be taken forward any further, and that is where you have the next generation sequencing technologies coming in. There were many technologies that came in, but some of them that have survived include your Illumina sequencing, which is the most common. 80% of the market share is coming from Illumina. And then you have what is known as the Iron Torrent. These are basically short range sequencers. The read length is around 150 bases. And then, of course, if you move on, now the trend is towards long range sequencers. And there are two techniques here that are very common today. One is nanopore, and the other is SMRT, or single molecule real time sequencing. Tomorrow we'll talk more about this. We will now conclude in another 5 minutes. It is already almost 1.5 hours. So I guess there comes a capacity of saturation. So I don't want to exceed that because that would also result in decreased interest. So the idea is that if you look at the basic steps in genome sequencing, you have to create a genomic library, split the genomic data into smaller pieces. Then you have to basically sequence these smaller pieces. So if you're using Sanger sequencing, you could split it to 1.8 kb. You could sequence the first part from here and the other part from here and put them together as 1.8 kb of sequence. So you have sequence of A, you have sequence of E, you have sequence of D, you have sequence of C, and you have sequence of D, right? Now, how do you put them together in order? So while fragmenting, you have ensured that there is some overlap between them, right? And overlap, so how do you use the overlap information? If the 3' end of this fragment has a unique overlap with 5' end of this fragment here, which means the overlap is only between these two fragments and not between any other fragment here, then you know that this fragment is going to follow this one. So now you can put this one first, this one as second, the D as second. Then you look at the 3' end of D and you compare with all other fragments to see if any of these have a overlap or similar sequence at the 5' end with respect to D, right? So whatever you find here, that is your next fragment, and so on and so forth. Then, of course, you have to do the assembly, arrange the reads in order they are likely to appear in the human genome. So this is largely wet lab experiment, creating a genomic library and sequencing. This is wet lab. But here comes the bioinformatics, right? You want to decipher the order of fragments in your DNA and arrange them, assemble them together. This is purely a bioinformatics task because you have the strings now, the sequence available, the partial sequences available, which may or may not have overlaps. You have to find out first which, take one particular sequence and compare with all others. The 3' end of this should have a unique overlap with the remaining any one of them. Any one, just one. It cannot be more than one. Otherwise, then again, you have this problem of which one is the next one. So therefore, this is bioinformatics challenge. And this is also, the time when bioinformatics until then was mostly about protein structures and saving the protein structures or 3D structures of proteins which are coming from X-ray crystallography. So bioinformatics, the beginnings is through the Margaret Dehoff protein data bank starting, right? And then of course, as the Human Genome Project came up, a lot of bioinformatics came into sequencing the DNA, arranging the DNA in the order it is likely to be in the human genome. Then 2001, this was the paper that came up in Nature. This is from the Human Genome Project. This one is from Science, the Craig Winter's Celera Genomics company. They published their own paper, but the two papers were published simultaneously in two leading journals of Nature and Science. And the announcement of the completion of the Human Genome draft was made on the same day. And this was basically announced by the then President of the United States of America, Bill Clinton, right? So and then, of course, the genome was not absolutely complete. I've already told you yesterday that the actual complete genome of the human DNA has only been sequenced now in 2022. This is because also there is a shift to what is known as long-range sequencers. And if you look at the Human Genome Project, it covered only 90% of the chromatic genome. It had 2,50,000 gaps and contained many errors in nucleotide sequence. And only in last year, 2021, we had the 20 years of the human genome. So Nature had come with a special issue in February 2021, celebrating the achievements of the genomic era and basically the achievements of the past 20 years. So you can see 2000, release of the first full genomic sequence of Drosophila melanogaster, 2001, publication of the draft sequence of the human genome. And then since then, the genome sequencing is now is not so much of a challenge today as it was in 2001. One was the technology, Sanger sequencing. It did not have enough parallelization. The other thing was that the when you're sequencing the first genome, you're standardizing your operations, right? You're basically preparing your SOPs, what worked the best. Once you had these SOPs in place, sequencing other genomes became a lot easier. So you can see 2004, you had the metagenomics sequencing technology coming up. Then you have HapMap project. Then you had several different sequencing technologies. And now, of course, genome sequencing is very easy, right? What you got was the reference human genome. October 2004, 93% of the human genome, which corresponds to 99% of the euchromatic portion of the genome, had been sequenced with an error of less than 1 in 100,000. This is what is the finished draft of the human genome, right? 2004. And then, of course, major findings. First, of course, we had an idea of 3.2 billion base pairs as the size of the genome. Then, average gene size in human genome is around 3,000 base pairs, and it can vary greatly. The biggest human gene is dystrophin. The biggest human protein is anyone? Biggest human protein is the biggest human protein is titin, right? Titin, titin. Then the total number of genes was estimated to be around 25,000 thereabouts, which was much lower than the initial expectations of having more than 1 lakh or 1,25,000 genes, because Drosophila had around 18,000. So this is something that came as a surprise, that the number of genes are actually very less. 50% of the genome consists of repeats, which is also a surprise. So a large volume of the report is of the genome is actually transposon-derived repeats. So therefore, that was also a big revelation. The human genome sequence is almost exactly similar, and if you compare two individuals, 99.9% is exactly the same. The 0.1% where it is different is what you can use for DNA fingerprinting, right? And these are basically specific regions like some simple sequence repeat regions and others where there are differences other than the normal SNPs. And then, of course, the functions are unknown for more than 50% of the discovered genes even today. A lot is left to annotation. So the idea is sequence is one thing, but if I give you 3.2 billion base pairs of just the ATGC, order of ATGC, it doesn't mean much unless you annotate it. What is annotation? Annotation means you're adding structural and functional information to a particular locus, a particular location, or a particular part of the genome. Chromosome 1 position X to position Y, gap DH gene. So this is the annotation, first level, right? Then second level annotation would be how many introns and exons are there in this gap DH gene. Third level annotation would be how many different types of isoforms of mRNA are formed in this region. Fourth level of annotation would be whether these isoforms are tissue specific or they are generic. Fifth level annotation would be the protein formed. What is the function of the protein? Which network it follows, right? Is there antisense RNA against the gene that can exercise post-transcriptional silencing? Is there other mechanisms of which the gene can be silenced? What is the promoter of the gene? Is there an enhancer somewhere else in the genome that can control the expression of this gene? What are the transcription factors that bind to it? So, there is a whole lot of annotation is limitless. The more our knowledge of biology increases, the more we are going to learn about biology, right? So therefore, annotation is something that is an unfinished business. So therefore, annotation is one thing that is the most challenging part now. Genome sequencing has become a lot easier, so much so that you can now sequence a human genome in a day's time. Then, of course, in the original draft, we found more than 1.5, 1.4 million single nucleotide polymorphisms were identified, SNPs or single nucleotide polymorphisms. Class, you're following everyone with me?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Okay. So then we move on and we talk a bit more. How is it arranged? All of you know, anyways. So there are 23 pairs of chromosomes. The nomenclature has been based on size. So chromosome 1, 2, 3, 4 until this is chromosome number 22. This is your autosomes, and then you have the sex chromosomes X and Y, and then sometimes mitochondrial genome is also added as a mitochondrial M genome or mitochondrial genome, which is independent of this and which is just 16.5 kb. And then, of course, you can also see that most of the part of the genome is euchromatic, except in case of Y chromosome, where you see you have more of heterochromatin as compared to euchromatin. Two-thirds of the distal part of Y chromosome is mostly heterochromatic. Large palindromes are present. We still do not understand why is it so, but that is how it is, right? Then if you look at the functional fraction of the genome, that is even more surprising. If you look at the total genes that are there, they are not more than 2% of the total genome, right? So what is expressed as proteins is just about 2% of the total genome, even lesser than that. For example, here it shows 1.1%. Another 4% is the regulatory part. Then you have 45% that remains heterochromatin. And then you have 6.5% that is basically your the 6.5% is heterochromatin. This one is the transposons here, 45%, and another 44% where you are not sure what it is doing, right? So there is a whole lot of dark matter in the genome of which we are not sure what it is doing, why is it there. So only about 1.5% of the genome encodes for instructions for synthesis of proteins. The functional elements in the genome can be classified into protein coding genes, which is this one here, non-coding RNA only genes, right? So these are basically your ribosomal RNAs and transfer RNAs. Regions of transcription regulation, this is this 4% that is basically for control of the genes. And then conserved elements not included in other categories. So what has also been seen is that while the genome, only about 1.5% of the human genome codes for proteins, but when you do an ultra conserved regions across the vertebrates, you find some 4% of the genome is conserved. So while we do not know any functionality to this, these are strongly conserved. And strongly conserved means there is some functionality to them which we do not know of today. So that is what is the fourth category. Conserved elements not included in other categories. And so therefore, there's a whole lot that needs to be understood even today. Repeat sequences are thought to have no direct functions, but they shed light on chromosome structure and dynamics, right? So as of now, we now know that repeats are very important in genome in the sense that they can serve as genomic novelties, can really take up novel functions. But there was a time when repeats were considered to be absolute useless, and they were termed selfish DNA, right? Or jumping genes. And most of these repeats are in the form of transposable elements or jumping genes discovered by Barbara McClintock. Now, what is important here is like this 44% question mark is, so for example, I mean, there is a recent example that can be given of, having known something for a very long time, but actual understanding of what it does came very later. For example, CRISPR-Cas. So the CRISPR as a repeat array was known to be present in bacteria way back in 1987. It was first discovered in 1987. It was only around 2005, 2006 that people started understanding that this is a part of a adaptive immune system in bacteria, right? So what can happen is you can have some information for a very long time, but you may not understand its true significance. And one fine day, suddenly it dawns on you, okay, this has been something that we had not been looking at. And then from there on, once you open this unlock this area, there is a whole treasure trove that comes up. So CRISPR now, 2028 is a Nobel Prize, and there's a whole lot of CRISPR applications coming up, not just in genome editing, also in terms of diagnostics and also in terms of kind of supplementing the protein product of a gene that is in faulty copy in heterozygous state, right? So the haploinsufficiency it is called. So there are many functions that come up, and sometimes you do not realize the significance of your finding to begin with. But now we know that there is a whole lot of DNA in the genome that it is not clear what it is doing, but it is pretty much there. And it has also been shown that almost 85% to 90% of the genome is transcribed. Whether genic or non-genic is not a question. RNA copies of those are formed, right? So there is a whole lot that needs to be discovered now. Then what was also seen is, how do genes evolve? So one of the most common methods of genes evolution is what is known as duplication. So there is, if you look at the globin gene, there is an ancestral globin gene which made into two copies. These two copies, because now we have two copies of the gene, one copy is independent to mutate, right? So therefore, over time, what has happened is this has accumulated mutations and become slightly different, diverse from each other, giving you now a distinct alpha and a distinct beta, right? And then alpha moves on to further divide several times, giving you almost 7 copies. Likewise, beta also, replicates many times and gives you again 6 copies. And these then basically still retain the original function, but they have a slightly different function. So you know that there is an adult hemoglobin and there is a fetal hemoglobin. So why should fetal hemoglobin be different from the adult hemoglobin? Anyone? Why should fetal hemoglobin be different from adult hemoglobin?
Fetal
Yes.
hemoglobin has higher affinity for oxygen.
Why should it have higher affinity?
Otherwise, it has to be the oxygen needs to move from the mother's hemoglobin to the fetus, right? If it's not, if it doesn't
Exactly. So that's the whole idea, right? So the fetus is not taking oxygen through the atmosphere. The fetus is taking oxygen through the maternal hemoglobin. So unless the fetal hemoglobin has higher affinity for oxygen than what is there in the maternal hemoglobin, it will not be able to pull oxygen from there. So therefore, over time, because it gives you an evolutionary fitness and evolutionary adaptation in terms of survival in the womb, therefore it has happened this way that you have two different hemoglobins in fetus and in adult, right? So what happens is basically what the idea I'm telling you is that a single gene copy first divides into two, gives you alpha beta, then they further divide, and then they give you some specialized functions. Some of them also may become pseudogenes. For example, the green ones here are pseudogenes. So they are basically now defunct copy of the original gene, right? So when a gene basically forms a copy, the copy can have two evolutionary pathways. One, it can evolve into a similar related function but slightly different function. Second, it could become a pseudogene. And what can also happen is that over time the divergence is so much that you may not be able to understand that okay, these two genes are were at one time related to each other and as basically have a common ancestor. So the idea is that because they have come from a common ancestor, and by accepting certain changes, the core of the sequence is still the same, and therefore these two copies can be aligned to each other. When you put them side by side, you will be able to see most of the nucleotides are similar, most of the proteins or amino acids are similar to each other, right? So that is basically, and the converse is also true. If two sequences are highly similar to each other and are having the similar function, they are likely to be coming from a common ancestor, right? So basically, a high sequence similarity is an indication of evolutionary relatedness, right? That is the whole idea of homology. Sequences that have homology because of duplication event, they are called paralogs. So now let's say you're comparing human alpha globin with chimpanzee alpha globin, right? So they have commonality because of the vertical descent. So these will be what is known as uh, orthologs, right? And if you're comparing the, let's say, the alpha and beta in human, they are paralogs of each other because they come from a common duplication event earlier on. Then again, the repeat content in the human genome is extremely high, 55%. Most of it is transposon-derived repeats, around around 50% of the total 55% repeats is transposons. Then you also have pseudogenes. These are duplicate copies of the original gene that have undergone mutations. More commonly, the nonsense mutations or missense mutations that render the protein non-functional. Then you have simple sequence repeats. So there is a motif, let's say ACA motif coming 10 times. So that is also a repeat. These are tandem repeats. Then there are segmental duplications. So these are large blocks of chromosomes getting duplicated out of errors in replication. And that can result also in basically a second copy of the original gene. And then, of course, further gene evolution. And then, of course, you have certain satellite repeats in the centromere, and then, of course, telomere as well. So these are your different types of repeats that you can find in the human genome. So this is some of the major findings. Now you could, because now you have several genomes available since the sequencing of the human genome, what this has led to is what is known as comparative genomics. So you compare genome of human to chimpanzee to mouse and see what are the differences, what are the similarities, and so on and so forth. You could also draw out general, the patterns of evolution through this. In humans, there are 3 times as many proteins as in fly because humans undergo a lot of alternative splicing. Although the gene number has not changed much, flies have around 18,000, humans at max will have 25,000. But the number of proteins is 3 times higher because of the extensive alternative splicing that happens in case of humans. Then humans share most of the same protein families with worms, flies, and plants. So in terms of families, in terms of functionality of the protein, there's not too much of a change. But the number of gene family members have expanded in humans, especially in proteins involved in development and immunity, right? And of course, then the human genome has much greater portion of repeat sequence as compared to other animals or plants. Now, what has this led to? 2001 as a watershed era in case of biotechnology, because we had conquered the biggest challenge of those times, sequencing the human genome, 900 base pair at a time, and sequencing 3.2 million base pairs, including the repeat portions that are there. So this was no ordinary task. It took 10 years, $3.2 billion, but it was done. And this led to a flurry of genomics programs, and a lot many genomes were sequenced very quickly. So for example, here you had mouse, dog, rat, chimpanzee, cow, marsupial, monotreme, birds, then woolly mammoth, Neanderthal. So all of these sequences came trickling by because you have standardized your protocols sequencing the human genome. The others were not so difficult to sequence because they were already done. One of the most complex genomes was already sequenced. And also what happened was there was advent of next generation sequencing, which made your task easier. The massively parallel high throughput of NGS made it a lot quicker with a lesser amount of money, and that was the again a key achievement that you had, right? And what this led to then is what is known as comparative genomics, where you could compare genomes of the same species, of different species, identify variations, and then try to see whether these variations are responsible for the phenotypic impact that you see, or the phenotypic differences in terms of normal versus disease that you see here. So that was something that was important. So comparative genomics, availability of multiple genomes, allows comparative genomics, comparison of intraspecies and interspecies genomes to understand our origin, evolution, and development of new biomedical applications. So this was again something important. Comparison of whole genome sequences from different species provides highly detailed view of how organisms are related to each other at the genetic level. So your molecular taxonomy and everything comes in. You could look at specific loci. Like the other day we were talking about. So if you're looking at prokaryote, 16S is one good gene where you can, if you sequence that and based on the variations that are there, you could identify your species up to the genus level. And if you do a whole genome sequencing, you can absolutely tell to the species, subspecies, and to the individual particular type of the species that you're looking at with whole genome sequencing. Conservation across different species suggests functionality of the genes. I already told you, when you compare all vertebrate genomes together, some 4% of it is very strongly conserved. Now we know that only 2% is 1.5% or 2% is accounting for genes. So there is another 2% that is non-genic and is still very strongly conserved, which indicates that there is some functionality to that region, which we do not understand today. Then minor conserved variations across different species suggest fine-tuning. So it is not that it is exactly conserved nucleotide by nucleotide. There could be minor differences at certain positions. that is more of a fine tuning to the specific requirements of the species in terms of the environment it is living in, and in terms of the geographical boundaries and everything. So it's more of an interaction between the environment and the species, and based on that, there are fine tuning of the same gene depending on what are the requirements. Then intra-species comparisons and variations therein correspond to disease phenotypes, facilitate biomedical applications. So this is where we are mostly concentrated. Comparison can be done at global scale. You could do what is known as global orthology, or at a local scale where you're comparing at the gene scale. So accordingly, there are two types of alignments. You have global alignment, you have local alignment, and we'll come to alignments in a moment now. But just to show you, this is your global orthology between mouse and human. Human chromosome color is gray, and wherever it goes and finds similarity in the mouse genome is shown here, right? So if you see here, human chromosome 1 is gray, and part of it is basically on chromosome 1 in mouse. Some part is there in chromosome 3 and 4, and then also there is yeah, so basically most of human chromosome 1 is a reconstitution of fragments of the chromosomes uh, 1, 3 and 4 in mouse, right? And Monod had once said very clearly that genome is a tinkerer. Instead of making new novelties, it will rearrange what it has to form something new. So this is a very elegant example of how human chromosomes are formed from the reassembling of the mouse chromosomes, right? So here you are, and I'm sure you know, chimpanzee, there are two chromosome 2s, chromosome 2A and chromosome 2B, which are fused together in human to give you a single chromosome. So that is why chimpanzee has 24 pairs and we have 23 pairs of chromosomes, right? So this is how these things work out. What you can also see as an evidence of this, the blocks moving around, is if you compare the human chromosome 14 and segment of this and find the gene order on mouse, the same genes in mouse, you'll find that all these genes are present in exactly the same order in mouse chromosome 12. So this is local conservation of gene order, which also is indicative of your basically the block movements and block rearrangements of certain fractions of the mouse genome to finally give you the human genome, right? So this is something that happens over times in evolution, and this type of conservation of gene order in related organisms is what is known as synteny. All of the chromosome has changed, but the gene order is the same. So basically, from mouse chromosome 12, a section moved into human chromosome 14, and the internal ordering of the genes remains the same, right? You're following class, everyone?
Yes, sir.
Okay, good.
Yes, sir.
So now we should get back to the practical. And just to tell you, because it took so much time and effort, what happened was it was also realized that the Sanger sequencing is not enough if we want to move to our goal of precision medicine or genomic medicine. And that is where you had the advent of the next generation sequencing methods, the second generation and the third generation. Some of these techniques have become obsolete now. For example, sequencing is not used anymore. Likewise, solid is also not used anymore. But virtual terminated sequencing and nanopore are used even today. And then we are moving towards the third generation, where we are now looking at longer read lengths, so that the assembly is not so much of a problem, right? So you want to have a longer read length so that you are able to assemble it de novo. You do not have to depend upon a reference for assembly. So these are your basically nanopore sequencer and your SMRT or single molecule real-time sequencing machines, right? So this is something that we need to keep in mind. So this is where the field is moving, and if you want to know more about this, you can go back to my, again, the YouTube channel, and there's a whole lecture. This lecture is something that I keep giving on and off, and the idea is every time I give this lecture, there is some new technology that has come. If you look at the 2018 lecture, I'll be talking also about solid and pyro sequencing. Now I don't talk about them because they become obsolete. So therefore, you can have a look at these lectures on my YouTube channel. Now coming to the alignments, right? So now I've given you a brief idea of how, because you have so many sequences available, you can do comparisons. And I've also told you, because genes most commonly originate via duplication of the original copy, and also these genes are vertically inherited. So therefore, sequence similarity is an indication of functional similarity. Also, it is an indication of evolutionary relatedness, right? So that is the whole idea of alignments here.